RNA splicing is a process that removes the non-coding sequences of genes introns from pre-mRNA and joins the protein coding sequences exons together in order to enable translation of mRNA into a protein. Introns are removed from primary transcripts by cleavage at conserved sequences called splice sites. A splice site can be located at either the upstream or the downstream part of an intron. For the former, it's called the 5' splice site or donor site, and for the latter it is called 3' splice site or acceptor site. Pre-mRNA is transcribed. It is quickly bound by several complexes called small nuclear ribonucleoprotein particles snRNP. snRNP contain RNA and protein, and it is responsible for splicing introns out of pre-mRNA. Then, snRNP bind to consensus sequence in a pre-mRNA at or near the intron exon boundaries. These sites contain nucleotide sequences that are shared by most pre-mRNA. The snRNP contain RNA molecules that can bind to these consensus sequence through complementary base pairing. Furthermore, to the snRP that have bound to the consensus sequences, other snRNP attached to other sequences within the intron. The snRNP come together into a large complex called a spliceosome. Once the spliceosome forms, the intron loops out. The spliceosome cuts the pre-mRNA at one intron exon boundary, where it leaves a reactive free O group on the exon. The spliceosome uses O to attack the other end of the intron, and in the process removes of the intron and joins the exons together, and mature mRNA molecules are formed. The mRNA leaves the nucleus and is translated into protein within cytoplasm. The intron that remains is quickly degraded. The snRNP remain in the cell and are used to splice introns from other RNA molecules. Recognition of introns in primary transcripts is partly mediated by detection of consensus sequences found at the junction of introns and exons, the 5 and 3 splice sites. U1 binds at the 5 splice site and U2 binds at the 3 splice site and the branch site, another region in the introns which is recognized by the spliceosomes. A trimer forms between the U4, U5 and U6 snRNPs. This trimer associates with both U1 and U2, bringing them closer together. This causes the intron to loop. After U1 and U2 are brought together, U4 is released. This release results in the formation of an active spliceosome to remove the intron from between the two flanking exons. Cleavage first occurs at the 5' separating intron 1 from exon 1. This loop structure is called a lariat. Cleavage then occurs at the junction between 3 and exon 2. U1, U2, U5 and U6 remain attached to the intron lariat and pair it to debonding enzymes located in the nucleus. The enzymes degrade the RNA and recycle the nuke. This process is repeated for each intron. When all introns have been removed, a mature mRNA is produced. Now the mRNA is ready to exit the nucleus and be translated by cytoplasmic ribosomes.